Welcome to our final night of the 2019 Biotech 201 session. Like boo-hoo-hoo, I am sure that after the deluge that we had to deal with last week, you were awfully happy not to have to swim your way here. For those of you that are in the space, those of you that are watching online, we're really glad to have you joining us. Thank you for being with us tonight as we wrap up our series. We have a lot to cover tonight. I say that pretty much every single week, and every single week it's true. We're going to talk about how we manipulate our microbiome, and antibiotics, probiotics, prebiotics, fecal microbiome transplants. But first, I want to say thanks to three groups of people that are the sponsors for Biotech 201 that allow us to do what we do each of the Tuesday nights. First, I'd like to thank JP and Orange Beard Productions. JP sits right over here in this back corner. All of you that are watching online, for those of you that are watching online, or if you in this room go back and watch later, JP is the reason why you can see everything and how you can get the handouts and everything. So JP, thank you. It is always a pleasure to work with you. I also want to thank the Jackson Center. The Jackson Center is a phenomenal host of our space. Yeah. And another round of applause for the cookies. And then I want to say thank you for all of the folks in this room that are watching online and elsewhere that are part of the HudsonAlpha Alumni Association. So this is our group. It's a dues-based membership group that actually provides funding that allows us to continue to offer 101 and 201 free of charge for everyone. And many of you in this room are Alumni Association members. Everyone will get an email in the next few days. If you're not an Alumni Association member, you'll get an email with the option to join the Alumni Association. If you are an Alumni Association member, you'll get an email that'll give you information about our members-only event. We do 2 members-only events every year, and this spring's members-only event is on the 28th of March, so a little away over a month from now. And we're actually going to talk about headlines that involve genetics, science fact or science fiction. So we're going to cover a whole host of things that you may see online or that you may have heard from your friends, and we're going to talk about if they're real or if they're fabricated, and what the genetics really tells us about that. It will be at the Probe Center, which is right next door. We have an education space in the newest building on the HudsonAlpha campus, and that's where we'll be for the Alumni Association meeting. So if you are an alumni member, you'll get that information. If you're not, you'll have the opportunity to join, and then you'll get information about that as well. All right. Let's stop and see if we have questions from our audience, either in this room or our online audience. Okay, so I will walk us through a little bit of our talk, and then I will stop and see if we have questions. We have a brave question. Yes, ma'am. About the glyphosate. So I started down that path, and then I got sucked into a probiotic storyline that took up my time. So I owe you an answer. So if you come find me at the break and make sure that I've got your name, I'll get you those details. So I apologize. All right, let's move on, and then I'll stop a little bit later, and we'll take more questions. So this week we're going to talk about how to shift our microbiome. And what is really exciting for me is that when we gave this talk 5 years ago, those of you that were here, there was very little that I could tell you. I told you a lot of don't go buy anything. I told you there's a whole lot. I didn't use this image, but I told you there's a whole lot down here and very little that we've actually got data on. 5 years later, that storyline has really begun to shift, and we'll share a lot of that information with you. You have a pretty big handout tonight. And the back several pages of that handout are a complex table that we're going to walk through some. But I actually wanted to put some data into your hands. We didn't have this kind of data when we did this 5 years ago. So we talked about these are the general ways that you can change your microbiome, or that you can change an individual's microbiome. Early life experience, we talked about the difference between individuals that are born through C-section and vaginal birth, that there are clear differences there. What those differences mean are a little less understood. We talked about differences in gut microbiome from individuals that are breastfed versus formula fed. And remember, I told you that many infant formulas are now trying to mimic the oligosaccharides, the prebiotics, and the probiotics that are found in breast milk to try to make formula even more closely aligned with what you would find in breast milk. We also talked about when you transition to solid foods. And then 2 weeks ago, we talked about this concept of what are you exposed to when you're young, the old friends. It was the healthy hygiene hypothesis and the old friends hypothesis. And does exposure to common bacteria actually protect you against things like infectious autoimmune disorders and allergies? I told you that diet, stress, and exercise are 3 things that we know change our microbiome. In fact, last week I told you those are the 3 things that you can say. Dr. Lamb says, reduce your Western diet, fewer high sugar, high fat, heavily processed foods. Exercise as often as you can near a daily basis, at least multiple times a week, and try to reduce your stress. All of those have been shown to impact your microbiome, especially your gut microbiome. Today we're going to talk about antibiotics, probiotics, prebiotics, and then this concept called fecal microbiome transplants, which are additional ways that we can shift the composition of what's in our gut microbiome. So let's start with antibiotics. So this is a table that shows you, what we understand currently about potential exposures to antibiotics and their impact on later in life disease. A fair number of these are in these 2 boxes, but there's a significant amount of evidence that begins to move some of these further up the chart. What we don't have an answer for is what is the impact of a single dose of antibiotics. I can't tell you what does that ultimately do long term. I can tell you some things about what it does short term and about the way that how long it takes the body to seem to be restored, but I can't tell you that taking a single dose or 2 doses or 5 doses over X years has this specific effect. The data still have to be gathered in a very precise way to give us that answer. But we do have some pretty significant insight that repeated early life antibiotic exposure may increase the risk of things like childhood obesity, type 1 diabetes, asthma, and allergies. We clearly know that there's a loss of microbial diversity when you take antibiotics repeatedly. The microbe composition of your gut becomes less and less diverse and begins to be dominated by a smaller number of bacterial groups. There's some evidence that if you are repeatedly exposed to antibiotics, so you are taking them multiple times throughout your life, that there's an increased risk of type 2 diabetes, and then very frequent infection in your teens and later in life is associated with an increased risk of Clostridium difficile. We'll dig into C. diff a little bit later, but the concept is if you are continually taking antibiotics, one right after the other, you are essentially doing the same thing as killing the grass in your yard. And when you kill the grass in your yard, what do you get lots of? Weeds. Because the grass crowds out the weeds. When you are on antibiotics repeatedly, you increase the risk that you are killing out your bacteria that would normally crowd out damaging bacteria like Clostridium difficile. So that's an issue that we'll dig into much more near the end of tonight's conversation. But there are at least fairly clear suggestions that lots and lots of antibiotics can be problematic. Now let me be really clear. There are times when the antibiotic is exactly what you want. So I am not saying that you should never be taking an antibiotic. I think we just need to be very cautious and judicious about the times that we do take antibiotics. So this is some of the data that's been compiled from the different studies showing you the impact of some types of antibiotics. Now, if you ask me, what about this antibiotic or that antibiotic, I'm not going to be able to give you that information. I can tell you the data that I found And we can't necessarily extrapolate from one type of antibiotic to the next because they have different modes of action. They kill different types of bacteria in different ways, and you may have been on different dosages or for different durations of the time. So it's very difficult to make one assumption about something you see on here and then extrapolate that to something else that you may be taking or someone else that you know may have taken. But oral amoxicillin shifts the microbiome, and that shift, that alteration, on average lasts 30 days. So you've taken a course of antibiotics, on average, your microbiome is going to take 30 days to recover. And in some individuals, it may take 2 months or more to recover. Some individuals recover faster. Some of that may have to do with their diet and if they are actually taking probiotics and what their gut composition was to begin with. So remember, everybody's guts have a slightly different set of bacteria in them. We are not all starting at the exact same place. Even if we all went through life with the exact same set of exposures, ate the exact same diet, we would have some differences in our microbiome. Large shifts have also been reported with Cipro, with oral ciprofloxacin. These changes were also persistent for several weeks, and some individuals, the restoration of their pre-antibiotic microbiome never happened. So it seems to be very specific from subject to subject, and that's also true for other antibiotic studies as well. Not everybody is going to bounce back in exactly the same way. This is interesting. The particular microbiome you're looking at comes back or responds in different times. The microbiome in your mouth, in your saliva, and in your throat tends to bounce back faster than the microbiome in your gut. Both of them are affected. Both of them see a reduction in diversity, but perhaps not surprising, the dosages that are making their way into your mouth are probably lower levels than the dosages that are being activated or hanging out in your gut. So your mouth and throat microbiome tend to bounce back faster than your gut. The other thing that is important to note is that antibiotics, they are a stressor of your microbiome. They cause this stress, and when your bacteria are stressed, they respond in different ways. The use of antibiotics actually selects for resistance among the bacteria that are surviving. So taking antibiotics, yes, gets rid of the bacteria that you're concerned about. Yes, also gets rid of bacteria that are beneficial, but it increases the likelihood that you're going to end up with bacteria that survive because they are resistant to the antibiotic. And if that ends up being the bacteria that are causing your infection that are now resistant to the antibiotic, that's problematic. Do you remember probably 2 weeks ago, we talked about this thing called horizontal gene transfer. The example I gave you was the individuals that ate the raw seaweed and the bacteria that lived on the seaweed, one of their genes that digest the seaweed was transferred into the gut bacteria of the people that continually ate this, that gene transfer. Horizontal gene transfer also happens with antibiotic resistance genes. And so you can have one type of bacteria that develops a resistance to an antibiotic, and it can transfer that resistance from one bacterial strain to another. So when you have something that selects, that increases the likelihood of resistance, that resistance can, it doesn't always, but can be passed to other bacteria in your system. That's part of the challenge, part of the issue. We had a question in the back, I think.
Yes, when you use the term gut microbiome, are you differentiating between the colon and the small intestine, or is that?
That is an excellent question. So when I use the word gut microbiome, what part of the gut am I talking about? Am I talking about your stomach, your small intestine, the entrance to your large intestine, or the end of your large intestine? I am collectively throwing all those together, which is incredibly sloppy, because many of the studies don't necessarily differentiate that as well. Which again brings us back to one of the challenges with microbiome studies is there is currently no gold standard for this is what we're looking for and this is how we're going to analyze it. So most of what we're talking about is large intestine, but not everything. Great question. We also have spent a significant amount of time talking about short-chain fatty acids. These are produced by bacteria in your gut they convert fiber into these metabolic precursors that other bacteria then turn into short-chain fatty acids. And short-chain fatty acids provide food for your colon cells. They are the major source of energy for your colon cells. And then they travel to other parts of your body, and they have all these other different positive beneficial effects. Antibiotic use drops the production of short-chain fatty acids because you are killing the bacteria that actually create short-chain fatty acids. And depending on which bacteria the antibiotic kills, you may be increasing the precursors. So you're turning fiber into these precursors, but you may have killed the bacteria that then turn these precursors into short-chain fatty acids. So you're throwing off the balance of where you're getting your energy from as well. Here are some options to think about to try to minimize the impact of antibiotics. Use only when necessary. You notice I didn't say never take antibiotics. You are not going to hear me say that. Use only when necessary. And then we also had a conversation the very first week. I got the question about the difference between narrow spectrum and broad spectrum. And I said, hold that until week 4. We're going to talk about that now. So the difference between narrow spectrum and broad spectrum antibiotics is a little bit of a semantic language, because some antibiotics are narrow in the fact that they only kill a specific type of bacteria. Others are broad strain in that they kill lots of different types of antibiotics indiscriminately. And then there are some in the middle that are more narrow than really broad, but more broad than really narrow. So how we classify that's a little tricky. Nevertheless, this is a general listing. I took the top 10 prescribed antibiotics last year and broke them down into whether they are classified as broad spectrum or narrow spectrum. And the consensus among clinicians is that you want to start with narrow spectrum wherever you can, because that's going to specifically target what is causing your infection. Now, that also requires that you have a fairly good sense of what's causing your infection. And if you just say it's a bacterial infection, but you're not quite sure what, that's where it becomes a little challenging. And that may be why broad spectrum antibiotics receive a fair amount of being prescribed. The paper that I've got on the screen behind me from the Journal of the American Medical Association was specifically looking at respiratory tract infections among children, and looking to see if the children that were given narrow spectrum versus broad spectrum showed any difference in how quickly they got better, or if they had any side effects or any additional symptoms. There didn't seem to be any difference between giving them broad spectrum and narrow spectrum. Now, the study didn't look at anything about their gut microbiome. They're just looking at why they are on an antibiotic. But this paper would suggest there isn't a difference between giving narrow and broad to treat the symptoms. And these authors argue that we should, as much as possible, be giving narrow spectrum antibiotics rather than broad. But as you can see from the table behind me, of the top 10, 6 of them are broad spectrum. So we are still prescribing a lot of broad spectrum antibiotics. Although recognize that language is a little iffy. It's very relative to to the impacts of other antibiotics. Yes?
That makes me, and forgive me because I'm looking at the list, I don't know which ones are actually in the penicillin family. So would the penicillin family be more the broad spectrum versus narrow?
So would the penicillin family be more broad spectrum versus narrow spectrum? So there are generation 1 penicillin family antibiotics, and there are next generation 2 and 3. My understanding is that the original penicillin was relatively narrow spectrum, and that it's the 2nd and 3rd generation that were actually identified because they had an even broader range in which they treated, which at the time was actually really beneficial because you could treat more things with it. We weren't thinking about some of the things we're thinking about nowadays. And that's one of the other issues. As I was digging through this, a category of antibiotic that operates in a certain way, there may be multiple subgroups within that broad category, some of which are narrow and some of which are broad. And I also didn't find that everybody always categorized broad and narrow in the same way. which makes it even more challenging. Great question. Other ways that you can try to minimize or reduce the effects of antibiotics on your gut microbiome is by taking probiotics at the same time that you are taking antibiotics. This is why your doctor may sometimes say eat a lot of yogurt while you're taking these antibiotics. Well, the yogurt contains specific types of bacteria, primarily lactobacillus, so you can help replenish that way, but there are a whole lot of other things that might not be in your yogurt. So maybe you would be taking other forms of probiotics. What type of probiotics would you take? So hold with me for a few more slides and we will get to that. But you can begin to try to reduce those risks by taking probiotic supplementation. Gary asked me 2 weeks ago if we were going to talk about phage therapy. And here's where we're just going to touch on it, Gary. We're just barely. But the concept of phage therapy. Phage are natural predators of bacteria. They are tiny. They are much, much smaller than a bacterial cell. They're a form of virus, and each phage has a specific type of bacteria that it infects, and it doesn't infect other bacteria. And it infects its genetic material into the bacteria, and it hijacks the bacteria to create more phage, and it kills it. So once scientists realized that there were these tiny phages that could quite effectively decimate very specific bacteria, phage therapy was used, not fully understanding what it was, early in, well before antibiotic use became widespread. So we weren't fully sure how we were doing this, but this was one of the early methods to treat infections. As antibiotic use became more common, phage therapy decreased in use, except in the Soviet Union, the former Soviet Union, where phage therapy was actually quite popular and remained popular. It's experiencing a resurgence of sorts as we begin to understand how selective it could be, and that it potentially could be used as a way to very specifically target a certain bacterial infection without impacting other different types of bacteria. There's a long regulatory approval road to get to this being widespread in use. But it is something that a lot of people are beginning to pay attention to and trying to dig deeper into. And you may have seen the paper that came out several weeks ago about a group of scientists, I think in Israel, that were using phage therapy to treat cancer. There's some interesting data behind that, but I really think, again, we're still down in these lower boxes. But stay tuned. We'll see where phage therapy ultimately takes us. All right, now let's dive in to probiotics. So the individual that I've got on the screen, Eli Metchnikoff, is really the father of probiotics. He is really the first to recognize that there were certain populations that drank soured or fermented milk on a regular basis and seemed to be healthier because of it, and began to advocate the consumption of fermented milk, and really grew quite a following with this concept that if you ate fermented products, brought beneficial bacteria into your system, that you would live longer, healthier lives. There's a really interesting history of probiotics. I had someone actually send me some information about the specific probiotic that Adolf Hitler took religiously in an attempt to remain healthy. There has been kind of a resurgence in our society about probiotics as we have shifted away from all bacteria are bad and must be destroyed towards embracing this concept of bacteria are good. We should make friends with them and we should bring them into our bodies. The official definition of what is a probiotic, according to the International Scientific Association for Probiotics and for Prebiotics and Probiotics, that actually says for probiotics and probiotics, sorry, that should be prebiotics and probiotics, says that the definition of a probiotic must meet the following things. the bacteria must be alive when administered. Now, there is some data that actually says that even dead bacteria or pieces of bacteria can have benefit. But the formal definition of probiotic is when you administer them, when you take them, when you eat them, when you drink them, the bacteria have to be alive. So if you're drinking something that talks about that it has inactivated bacteria, that is not a probiotic. Those are dead. I'm not saying they don't have benefit, but it's not a probiotic. Probiotics must have a health benefit. Well, that's a challenging one. How do you prove a health benefit, and what health benefit are you looking at? They must be administered at a specific dose, and they must be safe for the intended usage. Also kind of interesting, a specific dose. What is that specific dose? We'll talk about reading the labels on your probiotics and your probiotic foods in just a minute. And they must be a defined strain or a mixture of strains. In other words, the manufacturer must be able to tell you the genus, the species, and the strain. And that's important because, as we talked about last week, there are some genus and species where the strain, one strain is helpful and one strain is harmful. A few genetic differences in the recipe make the difference between whether it's beneficial or whether it makes you sick. So knowing the strain becomes really, really important. What's less clear about the definition of probiotic is it required to alter the composition of your microbiome? In other words, is it required to take up residence, or can it just wave as it passes through and does its benefit in transit? Does it need to live and colonize your gut? And do we clearly understand the mechanisms of action? In other words, do we know functionally what it is that this probiotic does? We have a whole lot of probiotics where the data seem to suggest that it is beneficial, but we don't fully understand the mechanism behind it. And right now, that isn't required to meet the official definition of probiotic. This is the shelves of my local grocery store. Probiotics are found in two places in my local grocery store. One, next to the fiber and the laxatives. Two, next to the vitamins. Everything in red is a probiotic. Let me just get out of the way and let you take a look at that. Everything in red is a probiotic. I spent a couple of hours at the grocery store. I am sure that the grocery store employees were wondering if I was like from a competitor, was I, price comparing, shopping, but looking at almost all of these probiotics and trying to get a sense of what health benefit they talk about, if they tell you strain, if they tell you dosage, if they tell you how to take them, and it is a wild, wild west. And this doesn't even include the food-based probiotics, like your yogurts and your kombucha and your kefir and all those pieces, which we'll get to in just a minute. This is just supplements. Although, let me see if I can find one of these. I'm not going to tell you which one, but one of these is dark chocolate embedded with probiotics embedded in it. As I got into that a little bit more, there were a whole lot of things about the dark chocolate and very little about the probiotic. So, I think it just was a way for people to feel good about eating some chocolate with a slightly higher price tag than just going to the chocolate aisle. But anyway, lots and lots. So my first point to you is if you take a probiotic, you need to be very savvy about what is in the probiotic that you are taking. And what does it tell you, and what does it not tell you? And you need to spend some time doing some comparison, which is part of why I've given you a whole lot of data for you to begin doing your comparisons against. This is big business. The global sales in the United States last year were United States, not global sales. United States sales, $2.4 billion in probiotics. And that's expected to grow over the next year and a half to $3.2 billion. And globally, it's projected that those sales will exceed $64 billion by 2022. We clearly are infatuated with the power or the promise of probiotics. But again, there is a wide, wide range of products on the shelves. Gary.
Who's doing research on these probiotics, like Consumer Reports or what have you?
There is no regulatory group that has provided oversight on probiotics. And that's one of the challenges. So there is no group that says this is exactly what has to be in it and that we're going to guarantee that what we say is in it is actually what's in it. Now, it is regulated. If it's marketed as a drug, it's regulated by the FDA. If it's not, there still is a congressional act that regulates it in terms of good practice in development. But there is no watchdog group. This ISAPP that I had at the bottom is probably the closest thing that we've got.
So it's got a science hook without the science.
It has a lot of science hooks, but the regulation around it is not necessarily scientific. And we didn't even talk about the probiotics you can get online, which is like 3 times this amount. And all sorts of nutritional supplements around that as well, and all sorts of different groups that try to tell you you should take these probiotics or those probiotics. I'm not here to tell you that you should not be taking probiotics, because I think we are now beginning to gather enough data that says under certain circumstances it makes sense to take probiotics. But I want you to be thinking about how you make the choice about the probiotics that you're going to take. And I can tell you, having now gone through the prep and reading the papers for this week, the way that I take probiotics is changing as well. All right. The World Health Organization categorizes probiotics in 4 broad ways based on what the probiotic is saying it does. So there's one type of probiotics that simply are live or active cultures, but it makes no claim of any health benefit at all. It just says enriched with live active cultures. The implication is that you will go, oh, that's a probiotic. That's going to be beneficial for me. I'm going to take that. But the manufacturer is simply saying there's live bacteria in here. Now, let me phrase that a little bit different. Made with live bacteria. It says that these are not harmful bacteria, but it's made with live cultures. The second one, yes, Hope.
I've had a question about buttermilk having any probiotics in it. Since it's pasteurized, is that enough to kill what's there?
If it's pasteurized, there should be no live bacteria in it. Doesn't mean there aren't dead bacteria in it, but there should be no live bacteria in it. I'm actually going to ask our moderators that are in the back room if they can do a quick search for me and just see if they can find anything about probiotics and buttermilk, and we'll see what they can tell me at the break.
There's a brand that hasn't been pasteurized.
That hasn't been pasteurized. And that's the whole issue of raw milk, which could quickly get us into a fist fight. So we'll step away from that. But yeah. All right, second option. A probiotic that's in a food like yogurt or a supplement like a tablet, and it actually says this is a probiotic. Not just made with live and active cultures, but it says this is a probiotic. It contains probiotics, but it makes no health claim. Now remember, a couple slides ago I told you that the definition of a probiotic was that it had a health benefit. This is just saying it's a probiotic, but we're not going to tell you that it's better for you, that it does something for you, but we're going to just say it's a probiotic. When you start looking at your labels, you'll begin to see this in ways that you may have been completely oblivious to. I was before this. Third category, there's probiotics in your food or the supplement, and they make a specific claim. Specific claim in a very general way, like reinforces the body's natural defenses. That's about as non-specific as you can get, but it still is making a health claim. This is good for your immune system. Promotes digestive balance. In other words, you aren't going to have a lot of gastrointestinal distress. Improves overall gut health. You can see how these are health claims, but they are very, very broad. They're very, very non-specific. And that's how you will very rarely see a probiotic with a claim that is more specific than that, because the data don't yet fully back that up. There's a few of them that you will see that specifically have claims because the research exists. They'll be very clear about that. But a lot of them are going to be this kind of general, non-specific, yeah, it's good for you. Go probiotics! And then a probiotic drug that specifically treats a certain disease. There are very few of these yet on the market, if any. 1 through 3 on here are regulated by the United States under the Dietary Supplement Health and Education Act of 1994, which essentially means they need to use good lab practice. They need to ensure that these are safe, but this is not a drug. It's a food product, so it has a totally different set of regulations. The fourth one, a probiotic drug, would have to go through clinical trials and FDA, full FDA approval, just like any other prescription medicine that you might take. Mark.
Do they have to do anything at all in Category 3 to show that their claim is accurate?
Do they have to do anything at all in Category 3 to show that their claim is accurate, where it says it has a health benefit? The bacteria that they use, the manufacturers, the labels that I've seen, say things like clinically shown to improve your body's natural defenses. So they are working with research papers that have been published that may be down here, but a research paper has been published, a study of patients, a clinical study, and that's what they're going to work from.
But it's not based on the product itself, it's just general clinical studies.
So is it based on the product itself that you are holding in your hand, or general studies on the same kind of bacteria? Most of the time, it does not have to be on the product you're holding in your hand. There are a small number of cases where there are specific probiotic products. VSL3 is one of them. That VSL3, which is a blend of probiotics, was used in the clinical trials. So you are buying the thing that was used in the clinical trials. Although what I understand is that VSL3 was actually purchased, the name was purchased by another manufacturer, and so what they were manufacturing under the same name was not the same blend of probiotics, but they've now been sued, and at some point they've agreed that they will stop that. So VSL3 at some point in the future will be the same blend, and it may already have made that change, I'm not sure. But again, you're buying rights to the name, not necessarily the blend.
Have you seen any research about the efficacy of refrigerated probiotics versus non-refrigerated probiotics?
Have I seen anything about the efficacy of refrigerated versus non-refrigerated probiotics? The data that I looked at, the International Society of, well, not on here, but that group, said that in many cases, as long as they are kept cool and not in direct sunlight, whether they're at room temperature or refrigerated, didn't seem to make a difference from the supplements. Now, if we're talking about yogurt, yeah, that makes a big difference. But if we're talking about supplements, the data, you don't want them to get hot. But they didn't seem to make they didn't seem to distinguish between the efficacy of what's in the refrigerator and what sits out on your shelf in a capsule. Other than the price point, there clearly is a price point difference there. Yeah, we got some savvy probiotic users in the audience that have been thinking through this. Oh, I just went through that. Let's just move us through. All right. Potential mechanisms of action. I'm going to dig into this lightly, and then we're going to take a break. We're going to do our break a little bit early tonight. But we've talked about multiple benefits of bacteria in your gut. So all these probiotics that we're talking about are gut probiotics. There are probiotics creams that you can put on your skin. Probiotic cosmetics is a really hot field right now. Makeup that is embedded with probiotics. I would be a little cautious about makeup with probiotics that I'm putting on or near my eyes, for example. or on my lips. And the jury is completely out on if these actually have any benefit. But they are a hot thing right now. Probiotic cosmetics with a really nice price point. But what we're talking about specifically is the mode of action of what you are ingesting. So we talked about that certain bacteria produce benefits to your gut, like short-chain fatty acids. So some of the probiotic benefits are that they are actually producing intermediates that are providing nutrients for your gut or producing extra vitamins and minerals. Another potential mode of action is that they actually are tightening up the connections between the cells of your intestines. So we talked about leaky gut, and there are certain bacteria that have been shown to reduce the incidence of leaky gut. They tighten up those connections, so you reduce that frequency. We also talked about that your bacteria have a role in signaling and stimulating your immune system. So some probiotics are thought to exert their effect by activating the immune system or helping the immune system do its job better. And then we also talked about the gut-brain axis. We talked about that last week. So some probiotics are thought to benefit your mood because they are specifically controlling signals or supplementing signals that are going to your brain. So the clear mode of action for any of these, in many cases, is not well understood. We've got some data that might suggest, but I don't think you're going to find a probiotic that clearly says this probiotic does this effect in the following 6 ways. So that's a place that science really still needs to catch up with us on. Yes? The statement is that you've done a lot of reading on bone broth healing leaky gut. Okay, so we are way out here. And at this point, my sister-in-law, who makes bone broth, is slapping me across the internet that I don't have the answer for that question. If I'm going to speculate, I would imagine it's that the bone broth, which contains a lot of collagen, is essentially acting as a prebiotic. It is providing the raw materials that the bacteria can then break down, turn into those short-chain fatty acids, and those short-chain fatty acids help reduce leaky gut. So I am guessing it is coming through that pathway, but that is purely a guess on my part. Yes, ma'am?
If a person is taking probiotics, like in number 3 that has these food supplements, and it's supposedly helping, can you get too much of the probiotic?
Can you take too much? Is too much a good thing? Too much a bad thing? Too much of a good thing, a bad thing? The vast majority of probiotics in your system do not take up residence in your gut, but pass by transiently. So on the one hand, taking more just simply means more of them are moving through. You're ending up with more of them in your stool at the end of the day. On the other hand, if they are actually producing proteins or metabolites, then too many of them could potentially be a problem. The data that I've seen has talked about about issues around bacterial overgrowth, that you may be creating an environment where your bacteria grow too well. I have not seen anything specifically that says you can take too many, but what I have seen is that numbers matter. So if you're taking probiotics that have a few million or a few 100 million live bacteria, the data is suggesting that you need to be taking things at a billion or a 5 billion or a 20 billion level. I think if you are taking something that's like 20 billion and you're taking lots and lots of them, the biggest damage that it's inflicting is to your pocketbook, because those are really expensive. Those ones that have like 20 billion live bacteria, that is not inexpensive. And if you're taking lots and lots of those, you are running through a lot of money. But I haven't sseen anything that says too much could be problematic. part of the challenge, let me be careful, let me be cautious, is again, remember, there isn't a lot of requirement. You can't always guarantee that what you think you are taking actually contains the bacteria that it says it does, especially when you are looking at the less expensive products. You want to be really careful about that so that you aren't taking something that you shouldn't. And the group of individuals that really should be careful about taking probiotics, period, are people that have compromised immune systems. So if you have someone with a compromised immune system, giving them probiotics to try to strengthen their immune system potentially could be problematic because you could inadvertently be promoting the growth of bacteria that they may not be able to control. So the data suggests, at least the what I'm seeing say be very, very cautious with doing probiotics with immunocompromised individuals. Hope, you asked about buttermilk. So all milk and cream are pasteurized, but buttermilk is made by adding acidophilus lactic acid bacteria. But the data, so it's made by adding a bacteria, but the data suggests that there doesn't seem to be any probiotic benefits from the fact that you've added that bacteria. And the question about the bone broth, my phenomenal moderators in the back said that it is the collagen and the prebiotic pieces that seem to be the issue around bone broth as well. Okay, have given you at the back of your handouts a set of tables from this document, the World Gastroenterology Organization Global Guideline on Probiotics and Prebiotics. That's a mouthful. You can actually download the entire report online. You don't have to go through a paywall. It's free for you to get to. I've given you the web address down here at the bottom. About 2/3 of the way through the paper, you actually get to the tables with the data in them. And before we go into how you interpret the tables, because we could go down a rabbit hole for days on this, I'm just going to equip you for how to look at this, and then we're going to pull back out. But the data, this is an analysis of multiple different studies on probiotics for individuals that have different diseases. And looking at all of the data and how were the studies developed, how many people were in the study, were these blinded, were they case control, were they prospective, retrospective, were they randomized, all those pieces. So this table that you've got, table 7, gives you a way for how to interpret what's called the level of evidence. So this is from the Oxford Center for Evidence-Based Medicine. This is a way that researchers and clinicians around the world look at lots of different kinds of papers and try to compare them across each other by saying, what's the level of evidence from this set of papers that gives me, is this strong confidence or is this relatively weak confidence in this answer? Number 5 is the weakest. Number 1 is the strongest. Number 5 is that there seems to be some logical science behind the mechanism for how this probiotic could theoretically make this difference. But it's not based on case control or clinical trial or randomized studies. Number 2 is I've done a randomized observational study. So I have split people into the groups that received the probiotics and the groups that received the placebo, and it was done in such a way that neither their physician knew who was in each category, nor the patients knew who was in each category. They didn't know if they were getting the probiotic or the placebo. So it was randomized and it was blinded. That is like the top level kind of study for these kinds of questions. But you see, it's number 2. It's not evidence level number 1, because evidence level number 1 is what's called a systematic review or a meta-analysis of multiple randomized controls. So one randomized control is good, one randomized study, but looking at the results of 4 or 5 gives you even more data. So as you look through the tables, and I'm going to show you the next one page of the table, as you look through the table, there are some key things that you want to pay attention to for how to interpret this. And again, I'm not telling you that This is what you need to go to the grocery store and be buying, but I want you to know that there is significant research from the last several years that actually, in many cases, ties probiotic use to better health outcomes for people that have disease. I could not give you that information 5 years ago. We didn't have the science. Now we do. That's exciting to me, to be able to say, yep, look, we've got something we can work with.
So is there a golden standard for a desired or objective gut bound? And can we measure the differential therefrom? Will we someday have a gut bound store? Are you going to
Ooh, that's a great, so is there a golden biome, a golden gut biome? This is the top of the class. And will we have some way that we can score it? So we have lots of ways that we can score it right now, but the problem is there is no core microbiome. That's one of the disappointments. There is no, this is the ideal microbiome, because it seems not to be based on what bacteria are living there, but based on what are the bacteria that are living there doing. So you and I could have two very different microbiomes in our gut, but if we've got populations of bacteria that are doing the same function, we would both be in good shape. So the golden microbiome may be based on a functional analysis, which is a whole lot more difficult than just saying, do you have this, 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 and this, and at what levels? Might there be a distribution? There probably is some sort of distribution, but until we have a standardized way of measuring this, we don't have anything of value that we could look at. You could take your gut microbiome at different times in the day, and you would get completely different answers. What's the right answer? What are we working from? How much sample do we need? What happened to what you ate 2 days in advance? And how long ago were you on an antibiotic? All those pieces go into that. So the hope is that 5 years from now, when we do this again, I'm going to be able to give you much better answers because we're going to have a better sense of how we score and standardize that information. Okay, here's how you're going to read this table. On the far left-hand side, it'll either be adult, and halfway through the table it switches to pediatric. So the first half is adult disorders. The second half is disorders in childhood populations. And then you're going to see what's the disorder. So for example, diarrhea. There are multiple kinds of diarrhea treatments. Are we talking about acute diarrhea, antibiotic-associated diarrhea, diarrhea from traveling? So on the left-hand side is what is the disorder that we're looking at. On the right, then the next category, what is this? Is this a probiotic, a prebiotic, or a synbiotic? A synbiotic is a combination of probiotic and prebiotic. The thinking being you are providing a beneficial bacteria and you're also providing the nutrients that help it grow and help it sustain. When you're looking at this, you're going to see a lot of words here. This is really complicated, and I'm sorry, there's no better way for me to give this to you than directly as it is. But what you're going to notice, like let's look like right here, Saccharomyces boulardii, and then CM I-745, or maybe that's a 1-745. In all these, Lactobacillus rhamnosus, GG. Genus is the first word, species is the second word, and strain is what comes next. All three of those are critical when you're looking at a probiotic. If you just see something that says Lactobacillus rhamnosus and doesn't give you strain, you don't have a good sense that you are actually taking the specific strain that the data is giving you information on. When I went back and started looking at all the kombucha that I'm drinking, more than half of the bottles of kombucha, more than half the brands do not give me, they don't give me anything but lactobacillus. That doesn't tell me a whole lot. That certainly isn't giving me the level of data that I need to be making decisions about this. So that's why you're seeing multiple bits of information, and those numbers and letters at the back are really important.
What are they again, Neil?
So genus, species, and strain. You're welcome. And then what's the recommended dose from the research paper? So that's not what's the recommended dose for everybody. It's what was the dose in the science that was being done. And you're going to see this CFU, this is colony forming units. This is the way that we measure how many live bacteria there are. So is this 1 billion bacteria, 2 billion bacteria, 3 billion? What is that? And then here's that key evidence level. that we talked about on the previous one? Yeah, thank you very much. There was a bug on me. It would have been really interesting if it had crawled like all the way up my face, you know. All the people online would be going, text Dr. Lamb, there's a bug on his face. All right, so. Wow, that was, and now, okay. Back to the slide. The evidence level, that's what we saw on the previous slide. What kind of study was this? Again, lower numbers are better. 1 is the best. And then what's the reference? Now, I didn't give you like the 40 pages of papers that you can connect the reference to, but because you can go online and get this for free, you can see what those references are if you wanted to dig deeper, and then specific comments that might be with that. So you have a whole lot of pages of this, which is a way for you to see what is the current level of data around probiotics and disease. Let me summarize it for you on this slide. Probiotics with level 1 evidence, that's the best top drawer evidence. Specific probiotics that prevent antibiotic-associated diarrhea. Reduction of side effects for therapy for Helicobacter pylori. This is a bacteria that's found in the stomach. It has some benefits, but it also seems to have an effect, a cause for ulcers as well. So what's the therapy around that, and what are the probiotics that can help with that? Prevention of post-operative sepsis for elective gastrointestinal surgery. Remission of pouchitis. Again, these are bowel issues. Lactose maldigestion. I can't break down lactose. I can't drink milk. I can't eat ice cream. I can't eat cheese. And then there's a whole set of pediatric gastroenteritis, hospital-acquired diarrhea, infantile colic, and then a gastrointestinal disorder. So we got 9. That's 9 more than we had. And there's a whole lot of other level 2 and level 3 evidence. But we're beginning to move in the direction that at least for these individuals with these disorders, probiotics have clinically proven benefits. So let's talk about healthy adults. Not just adults that have a certain disorder, but what about healthy adults? So this is a paper that I found that was incredibly helpful, a review of probiotic supplements in healthy adults, helpful or hype. And they did an analysis of all the different papers that had looked at probiotics in healthy adults. Pretty much everything we've talked about up until this moment has been associated with individuals with a known disease. Let's talk about now healthy. 45 studies were found that met good criteria for how they did probiotic studies. For example, did the patients know they were taking the probiotic or not? Because if you know you're taking it, there's a placebo effect that has nothing at all to do with what the probiotic itself is doing. So were they blinded, for example, to that? Were they randomized to that? So they looked at these 45 papers, and they looked at them for the following 6 categories. Gut microbiome colonization. In other words, did the probiotics actually take up residence in your gut? Did they strengthen your immune system response? Did they alter your lipid profile and risk of cardiovascular disease? Did they quiet gastrointestinal discomfort and bloating? Did they benefit the reproductive system in the female? And was there an effect on psychological health? And so we're going to dig into those quickly, keeping an eye on our time. So 45 original studies. Some of them looked specifically at one of these 6 categories. 15 were specifically asking the question, did the probiotic bacteria take up residence in the gut? And 14 of 15 said, yes, the probiotics seemed to somehow change the composition of the gut. There was a difference in the population of bacteria that lived in your gut. But the key was that 1 to 3 weeks after you stopped taking it, those bacteria disappeared. So they only hung out in your gut as long as you were continually supplementing them into your gut. They did not take up permanent residence. So they were transitional passerbyers. They were there while you were taking it, but they didn't stay if you quit taking it. Immune system response, I really found this one interesting. 3 of these studies looked at the impact of probiotics on the common cold. And they reported beneficial effects on reducing the incidence of common cold, the duration of common cold, and the severity of symptoms. That's a big deal. I just had the most horrific cold this year that I have had in a decade. Too bad I had not read this paper. And the specific ones that you can see are here in italics. Now here's the problem. This paper did not give me, it gave me genus and species, it didn't give me strain. So I've now got to go to the original papers, which I'm sorry I just ran out of time, to find what the original strain is to then go back and start comparing to my probiotics. But there is clearly accumulating evidence that probiotics are beneficial in reducing the incidence, the duration, and the severity of symptoms of the common cold. None of them found any impact on doing any of that for the flu. So something different in the common cold versus your response to the flu. And 10 of the studies actually reported that there were differences in the immune system of the patient, of the individuals taking the probiotics. Now the question is, are those differences in the immune system beneficial or not? And they don't have a whole lot of details there. It was really looking at things like your body's response to X, Y, or Z, which may or may not indicate strengthened immune system. There is no evidence from the studies that have been done to date that taking probiotics has any benefit on BMI, on fasting blood glucose levels, on insulin levels, or on your blood lipid profile. That doesn't mean that there's not, but the data to date have not found anything conclusive. So if you see something that says these probiotics are good for heart health, caution. Four of the 6 trials reported improvements. Now here we go. This is another, we have talked about all kinds of things in this course this month. 4 of 6 trials reported improvements in bowel movement, frequency, abdominal discomfort, and stool consistency. I'm just going to leave that there. And 4 of 5 trials looking specifically at the bacteria found in the in the vaginal tract. 4 of 5 trials found that taking primarily lactobacillus, different species of lactobacillus, increased the amount of healthy lactobacillus in the vaginal tract, which is thought to reduce the risk of vaginal infections. And then there was an analysis, a meta-analysis, so they went back and looked at multiple papers that had been done trying to identify if there's a difference on psychological health. Last week we talked about the gut-brain axis, and we talked about the effect of specific bacteria on things like depression and anxiety. 5 randomized controls suggested that there was a reduction in depression symptoms with probiotics, various species of lactobacillus and bifidobacterium, and 7 random control trials suggested that there was an improvement in subclinical. So you didn't meet the clinical definition of depression, but you had some sets of depressive syndrome or some set of anxiety. And several of these trials suggested that probiotics reduced the effect of subclinical depression and anxiety. Again, this is a relatively small number of trials, but they all seem to point to, yes, certain probiotics may make a difference in behavior. That was the difference between suggest and imported improvement was the wording specifically in the paper. And so I followed that wording because I wasn't really certain if there was a clear difference or not. But I didn't want to throw my own interpretation in. So all that to say, am I telling you that you need to now go out and take probiotics if you are healthy? I'm a lot closer to telling you that than I was 5 years ago. I'd say that with some caution and with thinking about specifically what you are taking and what else is in what you are taking, and you're not immunocompromised, I think there's a growing body of evidence that they can be beneficial. But just saying randomly, I'm going to take this probiotic is probably not the best approach to take. There needs to be some real savvy in thinking about what it is that you're going to buy.
So, number 1 was the
That is a great question. Why can't you keep the microbiome you're born with? Why can you keep that but not recolonize? You can recolonize, but you have to, it looks like you have to have had a major event that wiped out or drastically changed the microbiome in order for it to willingly take on new species that hang out. The microbiome seems to respond to differences in your diet and respond to differences in stress. It's very plastic, it's very pliable, but it also seems to be very durable. And that paradox between those two is something scientists have not gotten a good handle on. It's a great, great question. Specific strains matter. You need to pay attention to what is the strain that you are giving, and you need to know what strain you're looking for. Single strains versus blends. There's a lot of data that suggests that blends may be more beneficial because it is a blend from multiple bits of, you're taking multiple bacteria, not just a single strain, but that has not necessarily been borne out in every study. If you're looking at something that's got like 40 strains in it, I have seen no data that suggests that that's what you should be looking at. At some point down the road, to come back to your point about is there a golden microbiome, we may at some point be able to say, these are the things that you should take to ideally create your microbiome. But back to the question back here, right now, they do not seem to be taking up residence. They're just temporary passerbys. What's the amount of live cultures? In general, more is better. Where that more is, we don't really have a good sense yet. I've seen several recommendations of at least 1 billion or more. But again, I haven't seen clear data. I mean, you can see the doses on your tables. The doses are all over the place. So there is no clear sense of this is what the ideal dose should be. Buyer beware, because there is no overarching quality control. And I can't necessarily say that the more expensive it is, the better it's going to be. In general, if it's really cheap, that might be something that would make me kind of cautious. But I don't necessarily know that paying $100 for a week's worth of probiotics is any better than paying $30 for a month's worth of probiotics. This is one of those things that 5 years from now, I hope we have a better answer on. This comes right back to what you just asked. The structure of your existing microbiome may impact whether the probiotics you're taking actually do any good or not. They may be overpowered by your existing microbiome. They can't get a toehold, they can't do their job. And there are lots and lots and lots of studies, but the interpretation of them is really challenging, and that's why it's so hard for us to say, this study showed this, and so you can extrapolate that over here to you and your own individual health. Studies are done on populations, calculated in very careful ways. That doesn't easily translate to you and me sitting in these chairs or watching from home tonight. Read the labels. We're going to dig into that. Julie.
When there's an overabundance of yeast in the gut, do the probiotics help fix that, or does one need to take an antifungal like nystatin before the probiotics are?
So I have not, all right, so now we're at the edge of my knowledge, but probiotics are not getting rid of yeast. So if you have an overgrowth of yeast, you need to think about how you get rid of the overgrowth, and taking more bacteria is not necessarily going to help with that.
But the bacteria getting wiped out through antibiotics
and then the yeast taking over.
stuff, and then the yeast takes over, right?
Yeah, that's again back to, look, there's all this good nutrition coming through the diet, and I have no competition, so off we go, and we multiply because there's no thick green lawn to keep us in check. All right, read the labels. This is a label from a European paper, so be careful because there's a little bit of difference in the way labels are written in Europe and in America, but I really liked what's on this paper. Some key things that I want to point out to us. So, instructions that say, this one says store at room temperature and keep out of reach of young children. So they'll give you instructions about how you need to store them. Best before, so what date do these probiotics expire? These are live bacteria. Live bacteria will not live inside a capsule forever. So you've got to be looking for you've got to be taking them before their expiration date. And then next to over here, total active cell count. So what's the total number of colony-forming units. How many billion or millions of bacteria are in there? And is that number what it was when it was manufactured, or what it is at the time that you're likely to be taking it? Because there's die-off over time. So is it only telling you this is how many were in here when we first made it? Or is it saying that we guarantee that if you take it before the expiration date, there'll be at least this many bacteria in it? That's a really important distinction. What bacterial strains are present? And again, you can see here, Lactobacillus plantarum AB2. All three pieces, genus, species, strain. You want all of those pieces, not just contains live lactobacillus cultures. That does not tell you what you need to know if we're going to ultimately be able to know what strains are most beneficial and that we ought to be taking. This is a food supplement. This is not a food, this is not a drug, it's a supplement. So this is kind of the data that you're looking for when you read the labels. And you may be very, very surprised. So what about these kinds of fermented foods like kombucha and yogurt and kefir and sauerkraut and kimchi? So these are all foods that have been fermented, and they contain, in some cases, there are bacteria that produce lactic acid. So these foods often contain some sugar. There are bacteria that break that sugar into lactic acid, and then bacteria that ferment from there. And you can buy these foods in the grocery store. You can make these foods at home. If you buy these foods in the grocery store and they are pasteurized, there are no live bacteria in them. So just be aware that if you buy sauerkraut that's been pasteurized, and you like sauerkraut, that's great. But if you're eating it for the probiotics and it's been pasteurized, it's not going to do you any good. So let's talk about kombucha. So kombucha is black or green tea. Usually it's a little bit sweetened, and it's been allowed to ferment. And there are lots of companies that you can buy this from. You can also make this on your own. This is what is in my refrigerator at the moment. There are multiple different kinds of kombucha. When I sat down and started reading through the labels, I found that more than half of the bottles didn't tell me at all what was in them. And several of them taste really good because they are full of sugar. If you are taking a probiotic because it is healthful for health reasons, and what you are taking it in is packed full of sugar, you are negating all the benefit of the probiotic. Fermented food is fermented and has a fermented taste. If you try to cover that up with lots of other additives and flavorings, you are not doing yourself any benefit. So get used to the fermented flavor. Get used to the sour flavor. That's where the benefit is. Does heating affect that? Yes, heating can affect, because heating can kill the bacteria. So the level of heat can impact what probacteria are present. Now, I'm going to be really careful with this next one, because I know that there are several of you in this room that make your own kombucha. And I just want you to recognize that you need to be very careful when you do that, because you are creating your own colony of bacteria and yeast that grow in a symbiotic relationship. That's the SCOBY that floats on top. And unless you are able to know exactly what's in that SCOBY, you just need to be very, very cautious. I'm not saying that you shouldn't do it, because I know there are lots of people that are big, big fans of that. But you just need to be careful that you know what bacteria you are ultimately putting in your body. And you also need to note that some of these jarred kombuchas actually have added caffeine in them, which was a real eye-opener, because I drink my kombucha before I go to bed. And as I started looking at the label, I realized that one of the containers that I had just drunk had more caffeine, twice as much caffeine as the can of Coca-Cola that was also in my refrigerator. So, know what's in your labels. Pay attention to what's written on there. I thought, oh, I'm drinking kombucha, I am being healthy, I've bought it, so it's safe. Yeah, I need to really pay more attention to this. So the same is true. People that make your own yogurt, people that make your own kefir. the data that I looked at actually suggested that kefir, which is slightly fermented milk, has better data showing health benefits than kombucha. Kombucha is popular right now and it's fizzy. I like fizzy drinks. But the data seems to suggest you're going to get a better probiotic boost and a lot of other benefits from kefir than from kombucha. I'm not telling you not to drink kombucha. I'm just telling you here's what the data looks like.
There's a lot of sugar in kefir too.
There is a lot of sugar in kefir, unless the kefir is really sour. But if you're like, ooh, I like this one because this tastes fruity. Well, it probably tastes fruity because it has a lot of fruit juice and a lot of added sugar in it. Again, just be aware of what else is in your labels. Don't let the fact that it says made with probiotics that benefit your immune system or create a healthier defense, don't be fooled into thinking that you can then ignore the rest of what's on the labels. All right. Prebiotics. We're not going to spend a lot of time on this, in part because I'm almost out of time, but there's a lot less data around this. Prebiotics are non-digestible food. In other words, your normal cells can't digest them. The cells in your small intestine can't produce enzymes that break this down, but your bacteria in your gut can. So it passes through your small intestine and shows up in your large intestine as nutritious food to help the bacteria that are there. Here are some examples: dietary fibers, resistant starch, pectin, whole grains, inulin. Here's the key. The more we begin to understand about prebiotics, some prebiotics are beneficial to all bacteria, good and bad. They just provide the fuel source. Other prebiotics are what are called selective. We know that this type of prebiotic specifically benefits the growth of this type of bacteria. There's a huge debate right now in the field around, do you want selective or non-selective, and which one is better, and that answer hasn't been solved yet. And there are almost no clinical studies that actually say this prebiotic has gone through enough clinical data to say it is beneficial. We have a lot of suggestions. We know that fiber is good. Fiber is a prebiotic. But again, we are with prebiotics where we were with probiotics 5 years ago. All right. So the goal, where we're moving, is a personalized microbiome, where we have that gold standard and we know here's what you got, and then here's how you can alter that with your diet and with probiotic strains. We are nowhere near that, but that is a place we are going. All right, last set of stories. And it's 8 o'clock. I apologize. If you got to go, please, by all means, do, because I'm now holding you past when I contracted with you that we would do this. Fecal microbiome transplants are exactly what they sound like. You are actually taking the bacteria in the gut of an individual that passes into their feces, and you are transplanting it into the gut of another individual. So collectively, while we all kind of go, I will tell you that this moved from the last-ditch procedure to a widely practiced therapeutic option, especially for individuals with Clostridium difficile, with C. diff infections. This has been a game changer for people with C. diff, a game changer. And the donor, the person that's providing the donor stool, can be someone that you know, so just imagine that conversation. I've invited you over for dinner tonight because I would like to ask for a sample of your stool that I would then have put in a blender, liquefied, and then delivered to me either through an enema or a colonoscopy or a nasogastric tube. Here's a bag. But yes, you can choose your donor. In many cases, there's a really rigorous screening process. Or you can use what's called a universal donor, that's somebody that is already at a stool bank. Yes, those exist. Stool banks. We have blood banks, we have sperm banks, we have seed banks, we have stool banks. So, C. difficile is one of those opportunistic pathogens. It exists in your gut at really, really low levels, but when you've had a really heavy dose of antibiotics, it often clears out the competition and C. diff takes over. And it is really, really difficult for you to get rid of it, and it causes unbelievable gastric distress. Bloody diarrhea, huge pains, nothing stays in your system, and the only way to treat it is to take a stronger antibiotic. But C. diff can sporulate. It has this great defense system. It forms hard spores, and it can hibernate until you're done with the antibiotic, and then boom, it comes right back again. So fecal microbiome transplants were first reported in the 50s. Actually, there's a paper in China. There's a mention of it in China from like the 4th century BC. But in the 50s, there was the first mention of it, and then we heard nothing about it for 30 more years. People were like, this is the most unbelievable thing. This cannot be real medicine. This has got to be quackery. But 80 to 90% success in 2 to 3 days. And individuals that don't achieve success the first time, most of those achieve success with a second round. So this is a game changer in the way we think about, back to your question, in this instance, we are not just adding 1 or 2 bacteria, we are completely rewriting your microbiome and giving you the microbiome from someone else. That stays. That stays, and that stays and stays and stays. It's not something you got to go back in for an update, for a refresher on. I loved this quote. Although the Food and Drug Administration classifies fecal microbiome transplant as both a drug and a biologic, it is not comparable with any other drug in our system. There is no comparison of what this is. And because it was so successful in C. diff, researchers began looking at it for other sorts of intestinal disorders, like ulcerative colitis and irritable bowel syndrome, liver disease. It's even being looked at for autism. We talked last week about that study in mice that have a certain form of an autism-like form, that when they gave them a certain probiotic, many of the repetitive behaviors disappeared. The mental impairments didn't, but many of the social behaviors did. So there are people that are now looking at autism. Can you minimize some of the symptoms of autism with fecal microbiome transplants? Metabolic syndrome, and then this is a really interesting, relatively new one, patients that have blood cancers that have to have a bone marrow transplant. The process of destroying your own marrow, the chemotherapy, it wrecks your guts. So some of the early data suggests that many of these individuals, before they have their bone marrow destroyed by chemotherapy, if they save samples of their stool, and then after the bone marrow transplant, they have a fecal microbiome transplant, but it's their own pre-chemotherapy stool, that it quickly restores their microbiome in their gut. Yeah. Now, the challenge is many of these other tests have not been as successful as with C. diff. They've shown some success. They seem to show better success with certain types of donors, but they haven't shown the same success rate we see with C. diff. And that's probably, back to the point that we made earlier, with C. difficile, you have almost nothing in your gut. Your gut is wrecked. The microbiome community is completely destroyed and decimated. With these other disorders, you've got a lot of bacteria. They may be the wrong type. They may be in not equilibrium, but you've got them, and that may make it harder to work with. Okay, here's our last set of slides. There seem to be a group of people that are super donors. Their stool is incredibly successful for fecal microbiome transplants. So this concept of what makes a super donor, it seems to have a lot of diversity and some very key species. The Guardian, a public paper, got a hold of this and called these super pooper donors. Now, I saw Super Pooper, and being that I love lots of different kinds of musicals, I immediately went to Super Trooper. And in fact, wrote up an entire chorus for what this could look like. Because the background of the song goes, Super Trooper, and so you could have Super Pooper, all the way through. And the last line of that is feeling like I'm number one, and we could easily, anyway, but then I spun off in a totally different direction and rewrote Cher's 1997 song, Believe, and changed it to Yes, I Need My Microbiome, and it was great. It was phenomenal. I actually worked with we had a film group. We were going to do the whole thing, but then we ran into all these issues about it would be a public performance and we would have to violate all these copyright laws, and I really don't want to go to jail, so you'll just have to imagine how phenomenal that experience would have been. Here is our last slide. As we wrap up 4 weeks on the microbiome, We started by talking about how we move away from language about warfare with bacteria and how we live in peace with the bacteria in our body that are actually probably good for us, and recognize when we need to take medicines to get rid of the bad ones. The cautionary tale I would give you is we are moving from fear, from germophobia, to micromania, where we just embrace all things because probiotics must be the best thing ever. Be careful. Read labels. Don't just blindly step in and say, I'm going to take all these probiotics because they must be good for me. 5 years from now, and you've heard me say this like 20 times tonight, we're going to have a much better understanding of what this looks like. This field has been so incredible to revisit. I hope that you've enjoyed this interaction. The field is just unbelievable. I can't wait to see where we are after this. Thank you for being with me for the last 4 nights. It is a pleasure to be with you. Thank you. I'll be up here to answer questions. Megan Cochran, our genetic counselor, is also up here if you've got personal genetic related questions that you want to touch base with her about. Drive safe, everybody. Have a good night.